This is the help me video for the option test of component 3, done as the first key assessment. Right, option A, immunology and disease. So we're given some information about Lyme disease and how it's spread. Then we've got some definitions and some things to label. So this is largely the AO1 component. Remember, not a lot of AO1 on this. Endemic, so that's just a straight definition, and most of you got that. And likewise, putting your labels in here. Most of you were fine with that. There's no excuses for not getting those right. There's some tricky questions later on, but this is the stuff you should just be able to name. And again, define a term, antigen, and explain another term. So here we're looking for 2 marks, remember, 2 marks. So we're looking for the term. So an antigen is blah 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 blah, and then we want another bit of information about this antigen antibody complex. There must be something else you can say about it rather than it's when the antigen joins the antibody in the response. It needs to be more than that. Then we've got a graph part C, and we've got first exposure, so not at 0, the response for IgM. So not at 0, and then we've got the other antibody. You start at about 7 days. And the second exposure to the antigen isn't actually until sort of day 23, 24, that's that sort of time. And then C1, that's a straightforward explanation as to what happens after the second exposure. You're thinking about the type of cells that will be there after the first exposure. Part 2, a little bit more tricky. Lyme disease can persist in patients for many years, and the immunoglobulin M levels remain high. So this is a bit strange. You'd expect them to go down. You'd expect them to remain low, as they are on this diagram here. If they are high, well, what do you know about when they are high? Well, they're high on initial exposure to the antigen. So something's happening to this antigen so that the body gives a response that is after the first exposure. So the antigen could be changing, or the different forms of the antigen, and you just need to put that into the right words. So different forms of the antigen still giving that first kind of primary response. So try and think of the words that you're going to use there. Then we are given a procedure to test for these antibodies, and a bit of a convoluted rule. But there's the antibodies, and we're attaching an enzyme to them. Using the graph, explain why ELISA would not detect the immunoglobulin G antibodies. Most of you are okay there. You're going, right, well, that's where we're talking about on the graph, and we can't see any, so you just need to describe that. And the ELISA can produce false negative results for Lyme disease. In a false negative result, there is no colour change, even if the patient is infected with the bacteria. Suggest why. And the key thing here is, even if there are these antibodies in the plasma, the enzyme might not give a colour change. So quite difficult AO3 stuff, this. And you've done experiments with colour changes, and you've had problems in the past. So one could be a problem with this, this colour change and try and describe the problems that you've had with colour changes in the past. Another point here is, you look how much space you've got, and you don't really want to be filling all of this space, so you're looking to put a point every couple of lines or so. And then if you kind of give another point there, you know, a spare point. So don't spend half the space talking about 1 point. Another thing to think about is maybe something like here, you know, we are looking at different types of, I'm trying not to say the right word here, different types of antigen. So there's a slight clue there as to why you might not actually detect them in this test. Then you can think about some controlled variables that might not have been followed for this test. So how could this test have gone wrong with the operator not doing it right, or some conditions not being right for this test? So think about experimental conditions. And lastly, there's a point, you know, what happens if those antibodies have done exactly their job? Would they still be able to perform a colour change here if they are doing the job that they're meant to do in the body? Oh, quite tricky to get 5 there. Here's an experiment. You did this experiment in the lab using antibiotic discs, and we've got various different discs. Some have worked and some have worked better than others. State the temperature at which the culture should be incubated, and in the lab you cultured yours at 25 because we didn't want to encourage the growth of human pathogens. Here, well, this is proper science. This is proper testing of bugs that infect humans. So if we actually wanted to incubate this at the right temperature, well, it wouldn't be this safer 25, it would be something else. And explain your answer. Well, who gets infected and what's their temperature? The most effective antibiotic was E, okay, and gives you the diameter of E. You don't even need to measure it. Calculate the zone of inhibition, and it actually gives you the formula here. So pi r squared. You need to make sure you're giving r. 22 is D, the diameter, and a suitable level of precision. So a suitable level of precision is following what you've been given. So we've been given a 22 and a 3.14. So I'd certainly not give my answer to anything more precise than 3 significant figures. With reference to the image, so we need to look at the image, suggest what assumption is being made when making this calculation. So here's the bit of the image, and we're thinking about this zone here. It's near the edge there, so it's slightly flatter there, and it's a bit jaggedy there. But we're making an assumption about that, even though it's not quite perfect. You just need to put that into words. And that is the immunology.